So when the receptor binds to the ligand or signal, that triggers the recruitment or activation of the heterotrimeric G protein. And what it does is it triggers the GTPase subunit, the alpha subunit, to release its GDP, because it's in the inactive state at the beginning, and to bind GTP. And that turns it on. And in addition, by binding GTP, that releases the alpha subunit from the beta gamma complex, and that activates the beta gamma complex. And either or both of these subcomplexes, alpha or beta gamma, can then interact with downstream proteins and activate them. And they can activate a wide variety of different proteins. They can go and find a channel in the membrane and activate that. They can activate internal components, which I'll show you next. The GTPase here, the alpha subunit, has been activated by interacting with a receptor, and now it can go and activate other proteins. So in this case, it's activating this kind of brown protein, which is in the membrane. Eventually, the GTPase activity of this alpha subunit will cause it to hydrolyze GTP, and that will turn itself off. And so the timing, how long it stays on is a product of many things. This little gap here in the video that I tried to show that crashed everything shows that this can also be bound by a protein that's sometimes called a regulator of G protein signaling, but it's essentially a gap. It can promote the hydrolysis of GTP to GTP and turn this off. So there can be gaps in this kind of signaling as well. The beta gamma subunit also acts as a signal and can activate things. Often it activates membrane-associated proteins, like in this case, a potassium channel that allows potassium to flow out of the cell. And when this alpha subunit hydrolyzes GTP, that turns it off, and it also causes it to rebind with the beta gamma subunits and turn them off as well. Okay, so, whoops. Now things are going to get even more complicated, because when this trimeric G protein gets activated, as I said, the alpha or beta gamma subunits can go and activate other proteins. And one big class of proteins that they will activate are enzymes that make second messengers. What does that mean? That they're enzymes that do some chemistry in the cell that creates another kind of signal within the cell. One that's almost always shown in textbooks is adenylyl cyclase. So this is an enzyme that takes ATP and it makes it into cyclic AMP. And cyclic AMP is a very important intracellular second messenger. So it's called the second messenger because it basically is the messenger that tells the cell that the first messenger, which was the external signal, happened. When adenylyl cyclase is turned on, that creates high cyclic AMP within the cell. And eventually that gets cleaved by a phosphodiesterase enzyme. So it has a lifespan as well. So the signal persists until the cyclic AMP goes away. This is just an example of a neuron that is being acted on by an extracellular signal, serotonin, which is a hormone that makes neurons happy. It makes us feel happy and sated if we're eating and so forth. And the serotonin receptors on the surface of this neuron trigger the activation of adenylyl cyclase, and you can actually visualize the rise in cyclic AMP within this neuron using a fluorescent reporter that fluoresces more brightly or changes color when it binds to cyclic AMP. This kind of second messenger system can also greatly amplify signals. This would be an example in the eye, in a photoreceptor cell of the eye, which can be extremely sensitive to light. So if you have a photoreceptor that has the rhodopsin in its outer membrane, that rhodopsin, as I talked about in previous lectures, gets acted on by an extracellular signal, which in this case is light, and that induces a conformational change. A single rhodopsin molecule that's activated by a single photon of light will stay activated long enough to activate 500 individual G protein molecules, and that can lead to the production of, this is a different second messenger system than the one I already talked about. I talked about cyclic AMP. This is cyclic GMP, turning on the enzyme that makes cyclic GMP. This gets highly amplified. A single photon gets amplified 10 to the fifth or 10,000 cyclic GMP molecules that are hydrolyzed, and that triggers the activation of many, many channels in the membrane. So a single photon can then be turned into a pretty major signal that gets sent to the brain, and your eyes, if they're dark adapted, can detect even a single photon of light. So the alpha subunit is inactivated when GTP goes to GDP, but how's the beta gamma subunit inactivated? Okay, basically the same thing. So when the alpha subunit becomes inactivated, it rebinds to the beta gamma subunit and turns it off. So the hydrolysis of GTP turns off both the alpha and the beta gamma components. I'll just briefly mention that cyclic AMP, which is made by adenylyl cyclase, I mentioned that that's a second messenger, and one of the key ways that it acts is by turning on a kinase called protein kinase A, because it's dependent on cyclic AMP as a cofactor. So then this kinase gets turned on, and then it phosphorylates many, many downstream proteins. And one of the key pathways that this second messenger system is used in is to trigger glycogen breakdown in muscle cells to provide energy. So that's just another example of how a second messenger can then trigger additional events downstream. Active protein kinase A can also go into the nucleus. One of its targets can be transcription factors. By phosphorylating these transcription factors, it can turn them on, which will then trigger downstream gene expression. So it can change the protein components of the cell. But again, that's a fairly slow response because it takes time to transcribe and then translate downstream genes. This is a slightly even more complex case where the G protein, once it's activated, then activates this membrane-bound enzyme called phospholipase C. Phospholipase C is also a super important signaling molecule. It actually splits this inositol phospholipid, which is in the membrane. So this is the lipid tail, and this is the phosphate head group, which is called inositol. It splits it into two different second messengers, one of which stays in the membrane, that's diacylglycerol, and one of which goes into the cytoplasm, that's inositol triphosphate. Then the inositol triphosphate activates calcium channels that are in the membrane of the endoplasmic reticulum. That releases calcium into the cytosol. The combination of having high calcium in the cytosol plus high diacylglycerol at the plasma membrane activates another kinase called protein kinase C that then phosphorylates many, many substrates inside the cell. And one of the things that it does is it changes the actin cytoskeleton and causes cells to move and so forth. So this is a common but complex signaling pathway. If you take MCB133L, you'll do experiments where you use a chemical analog of diacylglycerol called a 4-ball ester that you can put on cells, and it will activate protein kinase C, and you can see the effects of that activation on the cells. That was G protein coupled receptors. The second major class of receptors that I was going to talk about are what we call enzyme coupled receptors. And these are receptors that are frequently interact with G proteins, but they don't just allosterically, meaning that they themselves are enzymes, or they interact with enzymes upstream of the G protein. So this is just an example of how an enzyme coupled receptor can work, where you have a signal that is actually a dimer or has, it's actually bound simultaneously by two molecules of the receptor, and that induces them to come together or dimerize. And sometimes what that can do is activate this receptor. For example, if this is a kinase, by bringing together another molecule, this kinase can phosphorylate this one, and this one can phosphorylate this one, and so they turn each other on by being brought into proximity to each other. And this is another example where you have a receptor that is by itself, not in an active conformation, but by binding to the signal, either two subunits come together or it undergoes some kind of conformational change and becomes active. It may become an active enzyme, or it may interact with another protein that becomes an enzyme. Again, many, many ways that cells do this. This shows kind of the general pathway for activation of receptor tyrosine kinases, which is a slightly more elaborate version of what I just showed you in the previous slide, where you have binding of a dimeric signal to activate a kinase. This is called trans-autophosphorylation, meaning that this kinase turns itself on, but one molecule can't turn itself on. It can only turn another molecule of the same kinase on. So by coming together, these turn each other on, they get phosphorylated, And these phosphorylation sites then recruit a whole bunch of other proteins to form a signaling complex within the cell. Hopefully you have all the building blocks. You understand that proteins can interact with each other. So proteins are always modifying behaviors of other proteins by physically interacting with them. These receptor tyrosine kinases are a very large class of plasma membrane receptors, and most of them work by activating a monomeric G protein called RAS. So RAS is like RAN, which I talked about before, except that RAN goes in and out of the nucleus, so it's free in the nucleus and the cytosol and controls nucleocytoplasmic transport. RAS is similar, except that it has this lipid tail that anchors it in the plasma membrane. So it's stuck to the plasma membrane. It gets acted on by these receptor tyrosine kinase complexes that activate, so that again, this acts as a GEF to promote nucleotide exchange and activation of RAS. And then active RAS triggers many, many different pathways in the cell. I'm mentioning this in part because RAS is one of the most frequently mutated proteins in cancer. I'll come back to this when I talk about the cell biology of cancer, but there are mutations in RAS that make it constitutively active, meaning that it becomes active even in the absence of this extracellular signal. And that's one way that cells can become mutated so that they can basically live and proliferate in the absence of the normal signals that they need to do that. So cancer cells can become kind of dysregulated by having these mutations in RAS. This video kind of shows how it works, but I'm not going to play it, partly because of the time and partly because I'm afraid of crashing again. So I will put it in the file, but it's worth watching. It just shows how the conformational change of the RAS protein is mediated by the binding of GTP and the hydrolysis of GTP to GTP. That causes a conformational change in the protein, which then affects its ability to interact with other proteins and activate them. So here's just an example of one of the pathways that are activated by RAS when it's turned on, something called the MAP kinase pathway. It itself is very complex. It involves a whole cascade of kinases that turn each other on. So this is MAP kinase, this is MAP kinase kinase, this is MAP kinase kinase kinase. I mean, this sounds insane, but this is how cells do it, and it's one way that they amplify signals. And I wanted to talk about something that was not shown in much detail in the book, but is actually a super important aspect of signaling pathways. For me, it helps me to think about how they might work within the cell. I've told you that activated G protein coupled receptors or activated receptor tyrosine kinases can act on many downstream pathways in the cell. And in fact, they act on overlapping pathways within the cell, which often lead to the activation of these different kinases, which have many, many downstream substrates. So you might wonder, well, like, how does a signal do something specific? If this can basically turn everything on and this can turn everything on, how do cells respond in kind of a specific way to a specific signal? Part of the magic by which that happens is that many cells express cell type specific scaffolds. So a scaffold, the simplest way to think about a scaffold is just an unstructured flexible proteins that have binding sites for a set of components involved in a particular signaling pathway. And by bringing these specific components together in space, the scaffold can basically shunt the effects of this signaling molecule down specific potential pathways, which can both greatly increase the efficiency of signaling because all the components are already together and they don't have to diffuse around and find each other, they're already there. And by binding to a specific subset of possible downstream effectors, a scaffold can basically determine which way a signaling pathway will end up affecting a cell's physiology. Posted a really nice review. It's a little bit higher level than the textbook, but it has really nice images that I think may help you guys to understand this kind of compartmentalization of signaling. This is the first figure in that review. You already know that organelles and also phase separation, which I talked about in the last lecture, are ways that cells can concentrate specific components and favor their interaction over their interaction with other components. So scaffolds are yet a third way, although they overlap, as I'll show you with phase separation. And the schmooing response that I showed right at the beginning of this lecture depends on this scaffold in budding yeast called sterol 5. it's called sterol 5 because when it's mutated, they can't mate. And it interacts with these 2 other sterol genes and also this gene called Bus3. And it brings them together into physical proximity and creates this specific signaling pathway. So in response to pheromone, the cells will undergo mating. And similarly, the mammalian MAP kinase pathway, which I just alluded to really superficially in the previous slide, is actually coordinated by the binding of these different kinases to the same scaffold. So they're all in close proximity to each other. So when a cell receives a signal, in this case, EGF, and RAS gets activated, that triggers a very efficient cascade of phosphorylation through this MAP kinase pathway that executes downstream effects. This just highlights something that I think is really cool, and maybe we'll talk about more in talking about infectious disease, but many pathogens manage to leverage scaffolds for their own benefit. And these are just 2 examples of scaffolds. In this case, YOP-M is a protein expressed by Yersinia pestis, the bacterium that causes bubonic plague. That was by far the most destructive infectious disease that humans have ever seen. And so when the pathogen invades the cell, it makes this scaffold, and that actually recruits 2 different kinases that are made by the animal cell that don't normally talk to each other, but by putting them together, the scaffold triggers the phosphorylation of PRK2 by RSK1. And so it basically alters the activity of internal proteins. And similarly, human immunodeficiency virus, HIV, actually manages to evade host inactivation systems by expressing this protein called VIF. And what that does, we haven't talked about this process of ubiquitin-mediated proteolysis, but basically what it does is it recruits this antiviral enzyme, which is pretty good at finding viral genomes and inactivating them. It recruits it to this protein complex that triggers it to get degraded. And so it basically disarms the cell that it infects by expressing the scaffold. And there are many other examples. And then finally, I just wanted to point out that scaffolds meet some of the key criteria that I talked about on Wednesday that promote phase separation. They're often unstructured proteins with binding sites for many other partners, so they're highly multivalent. And we now appreciate that one way that they can work is that they contribute to phase separation. So they actually bring together the components of a signaling system that they bind to into a biomolecular condensate, which further concentrates them and shields them from activities that can't enter that condensate and can thereby sort of insulate a signaling pathway from the influence of things that aren't in that phase. And this was just an example that I showed on Wednesday of this signaling pathway that leads to actin polymerization at the surface of these podocyte cells. And it's now appreciated that in addition to just scaffolding this whole pathway, these highly multivalent proteins also come together in a separate phase, and that greatly enhances their activity. Once again, I apologize for the slightly discombobulated. In this case, it wasn't my fault. This was just Zoom not playing well with Keynote or something else, but it's very frustrating, and it's distracting. So I apologize for that. Again, today's lecture is fair game for the quiz next week, but next week's lectures will not be covered on the quiz. Please get the textbook if you haven't already. And I will have office hours on Tuesday, and the GSIs will, I guess, communicate when they're going to have office hours next week.
Yeah,
yes, the textbook is free online, and you should definitely get it. It's a gift from the publisher, so you're ignoring a great gift if you don't get it.
Yeah, and we put out an announcement of when everyone's office hours are.
See you next week. Have a good weekend.